We are back in class, and I have the repeat of the first quiz to hand back at the end of class, and then we have these things to deal with this week. The first thing is our first exam is 2 weeks from today. Then we have an assignment due today. It's part of Chapter 7. Methods to learn about transcription factors. The book covers some of them. I put them in a list, and so there are questions to answer, and so email me the answer to those questions today. The questions are styled as though they're from an exam. Then the next thing is we have an estrogen receptor mind map video assignment, and that's due this Thursday. And so we just learned a whole lot of different things about the estrogen receptor, because this course requires me to pick and choose good examples that can be reused and repeated and really help appreciate the principles, right? And the key principle for us to start out with the estrogen receptor is it's a transcription factor, and it's different than MyoD because estrogen receptor requires a hormone. So it's a switch. And it provides a really good example of how we and every other cell on Earth can inherit a whole bunch of DNA and use it over the length of the life of the cell or organism. Is because one thing is there can be built-in delays and regulation in the timing, the location, and the amount of gene expression. And transcription factors play a big role in that. And so, we get into this thing where we have the estrogen receptor, but really not using it for several years, after we're born, right? So there's that. Then, just a short part of the first part of our class today, we'll finish up talking about Chapter 7. And so you should read Chapter 16. We'll start talking about that. And connected to Chapter 16, we started talking some about the PDGF and the PDGF receptor that's underneath the scabs that we have in our lifetime. And we have our last assignment before the first exam is that, and it's due in 1 week. And it's a big assignment. And so instead of asking you to, say, answer questions that are embedded in the assignment, like the one that's due Thursday, or like the other assignments you've had where I'm like, make some drawings, send me pics. I'd say, send me all your notes, because part of this is to also try to encourage you to take notes in class and during the assignment. And there are really two purposes for notes. One is to help you learn the material. So we talked some before about that. This is very physical, and in general, STEM topics are physical topics, and they can be depicted in the real world. So even though it's talking about molecular and cellular biology that are too small for us to see, in case of molecular biology, even with microscopes like that, can't see it directly, yet we can depict it in big drawings and stuff like that. And so we want to take notes because of the physicality of it, but then also your notes become your study guide. Students sometimes ask me, am I going to hand out a study guide? Any kind of review, and the answer is that's all I ever do, is I'm desperately using what limited class time I have to try to provide that. And you should be scrambling to create that study guide. That's what it is, right? Like that. So when it's time to prepare to study for the exam, you could go back and just watch all the videos again. That would take some time. Or you could go back and read all the chapters again, right? But there should be an intermediate thing, and it's what you're creating as trying to learn the material. You're also creating a tool that you can use to review and grow. So you're supposed to send me pictures of all the notes you have, and they'll be written, they'll be drawn, they'll be labeled drawings, and they should be big enough for you to remember those notes on the exam. Learning and exams are all about recall, and what you recall is either my voice from class or PowerPoint from class or the book or the images that you're producing, and if you make them big, they're more memorable, okay? So that's where we're at, okay? So what about this exam? So we all know what it's gonna be. It's gonna be 50 two-point questions. In kind of a crowded classroom, so I'm gonna have assigned seating. I'm used to a bigger classroom where we can make some space, right? So I'll just make some tags and you'll come in each of our exams and find a place to sit. So you don't end up sitting, next to somebody, right? You might be next to somebody, but it will be some random person like that. Because we gotta be, we'll try to spread out, so we don't have people just looking at other students' exams. And then we'll wanna get it done, right? So I've talked about that. So don't simply start writing the question again, but get to the answer, but also use as many words as you want, to explain to me what the answer is. I've talked about that, and because we have limited time, and I don't want somebody to run out of time. And I've written exams where if a student stays on task, everybody can finish it up like that, okay? Because there's another class in here after our class, so we'll have our time plus a few minutes, 5 or 10 minutes, and then we got to get out of here like that. So that's what we're getting ready for. There are 3 exams in the final exam, and all of them build up on each other. So for our second exam, you're not going to have to go back and study everything from the first part of our course, but it's stacked up. So after our first exam, I'll have a video saying, answer key, and you really want to go back through that and just really try to build up. And then our final exam covers the whole course like that. Okay, so that's where we're at, and our exam's going to cover those 3 chapters like that. So I've talked about that before, and I should just pause here and check in if anybody has questions about anything at all. Reminding you that the first time I taught this course, that's all I did, is I came in and said, this is what you should be doing now, let's answer questions. So, I just move on. If somebody, if there are any questions from chapter 7, I'm like, okay, I'm done talking about chapter 7, but because I figure, you're looking at that, and a small number of the questions on each exam will be from our reading, stuff that we just didn't get to cover. And it'll be something important from that chapter, not a trivial, footnote or, little word like that. Yep. So does anybody have any questions about anything at all? And another thing, to mention is just, to kind of be upfront and kind of help you anticipate, right? Everybody understands that in USM, biochemistry is a really hard course, right? Students really struggle with that. So this is biochemistry plus genetics. It's hard. But all of it, right, when we were talking about the mechanism of action of those transcription factors, right? MyoD helped get the TBP there to help assemble the rate-limiting step, assemble the pre-initiation complex. Lots of transcription factors do that. They help assemble that pre-initiation complex, including getting RNA polymerase to the scene, because they're tethered. They're bringing stuff locally. Then HSF, heat check transcription factor, fantastic physical story of it, right? There's already arrested polymerase sitting 70 nucleotides down the transcription unit, just paused. Like that last person to hand the burgers out at the shack, just literally, you gotta speed that up. And so HSF directly releases this arrested polymerase. Great example of Tues, because that heat check transcription factor is emergency first aid for the cell. So, great example. However, lots and lots of genes have just normal everyday genes and so forth have arrested polymerase as the rate limiting step. And then the estrogen receptor, super awesome example, where it turns out the DNA is always wrapped up in these histone octamers of nucleosomes and more, wrapped up, and kind of can be stored away in heterochromatin or opened up into euchromatin. And so that's what the estrogen receptor does. And other of these nuclear receptors, So just good, accessible stuff. It's just, just a lot of it, right? And so we got to organize it. Big part of this is organize your learning. Okay, great. So then just to loop back over this stuff, we could talk more about it. But these are the examples we talked about, given those examples I just mentioned. And then when we got to the estrogen receptor, right, we said, remodeling chromatin is a big story. And our book unpacks, no pun intended, lots of ideas about chromatin and chromatin remodeling. And I said, we're gonna, in our course, especially focus on 3 kinds. And we talked about histone acetylation. There's lots of stories about histone acetylation and deacetylation. we know one, estrogen receptor. Then nucleosome phasing, there's lots of stories about that in regulation of gene expression, is positioning those nucleosomes to make sure they're out of the way. And we know one, GAGA factor, and its function and the heat shock response. And then DNA methylation, we're not going to get a specific example other than that DNA gets methylated in differentiated cells. There's lots of examples of that, so we'll dive into that. We haven't really talked about that specifically. So that's where we're at, is we want to talk about DNA methylation just for a little bit. And so histone acetylation generally increases gene expression, so that's where we get that example, and we use estrogen receptor specifically. And we go back to our original videos, and that's just DNA, right? And it's depicted there, the clay is just DNA, but it's always wrapped up, it's always packed away in the chromosomes in our cells, and it's got to get unfolded. So, nice stories about histone acetylation and deacetylation. Something that I didn't mention yet before, that I'll put right here, is that these are, there's the estrogen receptor. In this case, the figure is actually talking about a different nuclear receptor, thyroid hormone receptor, and but it's a nuclear receptor, and we could say it's the estrogen receptor. There would be estrogen, and so then these are these enzymes, the histone deacetylase in the absence of estrogen, and the histone acetylase that get exchanged when estrogen shows up. And you see how these enzymes, they're really serving a role in transcription. So we could call those transcription factors. And remember how we emphasized that MyoD has a DNA binding domain and then a transacting domain. And the estrogen receptor has a DNA binding domain and then a ligand binding domain and then transacting domain, but it's got a DNA binding domain. It's going to bind to that enhancer. And then that heat shock transcription factor, that's that three-handed climber. The hands are DNA binding domains. It's going to bind to it, right? So we have transcription factors that we mostly talk about that have DNA binding domains that bind directly to DNA. These enzymes are transcription factors that don't bind to the DNA. Yeah? And so there's a general term for those. Those are called co-regulators. And people rarely use that term, but it means a transcription factor that does not have its own DNA binding domain. It's going to bind on the back of somebody else. Okay? The reason people don't use co-regulator very much is because they either call it a co-activator or a co-repressor. And so that just means co-helper, co-repressor. So this is a great example also because we get those two terms. Histone acetylase is a co-activator. And histone deacetylate is a co-repressor. So, just to add those additional terms in there because that's often talked about in studying transcription and regulation. Okay, all right, great. So, we talked some about that example, and then in that example, I just left a couple slides in because we just, stacked up that example. We leaned into the cancer biology. We learned our first cancer therapeutic, very significant, right? Somebody that is an ER-positive breast cancer patient can get this drug and it will help them because it's an antagonist of the estrogen receptor and the cancer is growing in an estrogen-dependent way like that, and so it can block that. And then we learned, there's also this concept of an agonist, and so BPA is an agonist. That's what its function, its disruption function is. It's called an endocrine disruptor. So, we drove into that a little bit, and we got this example of these transcription factors that require a ligand. And we include all the nuclear receptors, and that's in that video some about the estrogen receptor mind map. As this superfamily of nuclear receptors in vertebrates. And in humans, there are 48 of them. And they include the estrogen receptor, the testosterone receptor, the glucocorticoid receptor, the thyroid hormone receptor, vitamin D receptor, this thing called the peroxisome proliferation activator receptor, and several others. So just super significant families, and people call it a superfamily because they're so kind of different. And so here's their overall look. They all have the DNA binding domain, and it's a zinc finger binding domain. They all have a ligand binding domain and divides a divide of different ligands. Some of them bind hormones, some of them bind other kinds of molecules, and then they have that transactin domain, and the transactin domain binds to a co-regulator, either a co-regulator that's a co-activator or a co-repressor. So he talked some about that, talked about this superfamily, talked about how they're paralogs, and then I didn't really show and stop at this slide, but paralogs can look really similar. So it's weird to think about, but a screwdriver looks really similar to a shovel. It has a handle and it has a blade, and they're used for very different purposes, but you can see how you could evolve a shovel out of a screwdriver. Make the handle longer, make the blade wider, quit using it to screw stuff in, just use it as a lever like that, right? And you can use a screwdriver as a lever already, right? Sometimes you gotta pry something open, you like get an old screwdriver, get the lid off this can of paint. So they're still paralogs because you look at them and they're like, these are remarkably similar. They have the same overall structure like that. You're like, these are all super related. And people can easily make like a evolutionary tree of the nuclear receptors, of which, were the ancestral ones and the drives and stuff like that. Yep. So they involve remodeling chromatin to activate or repress gene expression. Then this is just that HSF and the GAGA factor, phasing the nucleosome. And then here's just a couple minutes of the third example of remodeling chromatin. So changing it around, and it's DNA methylation, and in general, it represses gene expression. And there's a lot of enthusiasm in the, in biology students today and in the general public, because it's a way of altering DNA sequences by methylating the Cs. And there's a lot of different stories of how that what the relevance of that is for the cell and for life. And we probably will only talk about one example, and that is in cells that differentiate, it's very common for them to methylate genes they're never going to use. So that's our good example, and it's going to shut down expression. And here's this. You don't need to know the structure of this, but just to see it once, it literally means add a methyl group to that position of cytosine. And it's just cytosine, and it turns out that that methylated cytosine starts to look a lot like thymidine or thymine. And so it's possible to have a mutation in methylated C that makes it turn into a T. And so it turns out that cells don't really want to methylate Cs. They want to be careful because it makes it vulnerable to a mutation, a nucleotide conversion. And so, just to add a little bit more to the story of methylated Cs, in animals, the C that gets methylated is almost always right next to, in front of, in the DNA sequence, a G. And so people refer to that as a CPG. And that's because the cell is trying to be really careful not to methylate C, because it sets it up for DNA damage. So let's restrict it so that our DNA repair mechanisms can look at this, and if they see a C next to a G and there's a mutation into a T, the cell will say, that's probably an issue. And as a result of that, there are regions in the human genome that people refer to as CPG islands. And that means there's just a surprisingly high number of CPGs in that region, and the region is in the cis regulatory region of genes. So when we talk about DNA methylation, we're really especially talking about regulating transcription and repressing, decreasing the activity of transcription, and mostly at these CPG sites, mostly at these CPG islands. And so that, you don't need to see that carefully, but it just shows that there's a very non-random distribution of CPGs because of their concern about mutation. Just a detail. And for us, in our course, we'll probably limit our example to CPGs and CPG islands and DNA methylation and gene repression to this thing of differentiation, that when a cell differentiates, it's just going to methylate those. And you'll see it's going to come up in a very different setting where we're like, this, all those are methylated, the muscle cell can't suddenly become an eye cell or a blood cell. It's got all the genes, it's got genomic equivalence, it's even got the insulin gene, but it's never going to express the insulin gene because it's methylated, like that, okay? All right, so that's that. And with that, that's all I wanted to say about Chapter 7. I'm happy to say lots more about Chapter 7, but that's Chapter 7. That's transcription and regulation. Remember, our course started, even before our first class meeting, I said, let's watch these videos about muscle differentiation. That's Chapter 7 stuff, right, like that, okay? So that's a big part of our course, all right? And now we're going to go on to this Chapter 16, and we go on to the fourth section of our book. And I think the fourth section might just be called cancer. And remember, I teach this course as if it's got a title, Molecular Mechanisms of Cancer. And there's lots of reasons for it. There aren't any other cancer courses at USM, and so we're going to teach molecular biology. What should our examples be? Well, not all of you have taken immunology, not all of you have taken neurobiology, not all of you have taken developmental biology, not all of you have taken animal physiology, right? What can we use as our examples? So, let's try to use something that's very useful, that's just maybe one examples that you haven't really run into. And that's the whole section of this. So just to look ahead, I find this back here. Our second exam is almost certainly going to be those three chapters. And so the flow is going to be like, cells communicate a lot. And in animals, the growth of cells, the normal growth of cells, like the healing that goes on under a scab, is regulated by cell signaling, cells communicating with each other. So that's the normal growth control, and mutations in the proteins that play roles in those growth pathways, those could lead to cancer. And we have one example already, RAS. RAS is involved in growth of cells, like that, right? So this chapter follows really naturally, like that. What's something that cells do when they get signaled? And one thing they do is they go through the cell cycle, right? So in cancer biology, we go right to that, but that's also our normal health. And we learn about that, and we learned that, cancer is a story of cells growing out of control. But then it turns out we'll run into some examples where we see that normally cancer cells get eliminated. And one way they get eliminated is the cell, the cancerous, precancerous cell, just kills itself through programmed cell death or apoptosis. So there's another thing that can really contribute to cancers, and that is blocking a cell's ability to die. And so we go right into that topic and run into a whole bunch more. And then the 19th chapter is kind of pulling it all together and talking especially about some therapeutic mechanisms that are all targeting, drugs that target things like RAS or MYC and so forth, like that. So that's probably going to be our second exam, like that. And then we'll go back into earlier chapters in our book and we'll talk about these. And it'll be nice because we'll have like good examples to look at those earlier chapters. So that's where we're at. Here comes chapter 16. Okay, so what we want to do is kind of get a big picture. And so cell signaling is about, we'll just draw two cells, but these would be two types of cells. And one of the cells we could talk about would be a cell in an ovary. And so some of the cells in the ovary are cells that make most of the estrogen hormone. And they release it into the blood, and it floats around, and there are cells that have the estrogen receptor, and that can stimulate them. So that's what we're talking about when we talk about cell signaling, is one cell, and it's really a population of cells, a group of cells, makes a molecule they release, and that's a signal, and other cells receive a signal. And this would be called, in the most general sense, that's a source cell. And then this would be called a target cell. And the target cell is going to receive this signal and it's going to make a response to the signal, right? So the estrogen receptor is going to activate genes like that, okay? And the response is called the cellular response. And there are really, and lots of time, and there are really two types of responses a cell can do. And lots of times it does examples of both of them. And so for cellular responses, one kind of response is called genomic response. And that's referring to the genome, and it's referring to new gene expression. And we'll especially use examples of that, where the target cell is going to change its expression of genes. The cell response is going to lead to it. Now, the cell response could be like change expression of genes and go into the cell cycle, right? Because that's what MYC does, and MYC is activated, expressed by that phosphorylated ALK, that's a target of PDGF receptor RAS signaling, like that. So that's an example of a genomic response. We've got to change gene expression. And then the other is just really easy because it's a non-genomic response. So something the cell does that doesn't require changes in gene expression, and that's it. And often the target cell does both. It does both genomic and non-genomic responses. The only reason we bring that up is because it's true, and also we just in our course super emphasize genomic responses because we super emphasize transcription regulation and the cis regulatory region and transcription factors like that. And that's one reason why the estrogen receptor is such a sweet example for us, is because it's just very simple, where you're like, and then the estrogen estrogen is called a lipophilic molecule. It's very hydrophobic. It's moving around in the blood, but it's rarely just floating on its own in the blood. It's stuck to something else because it's very hydrophobic. That means that it can actually go right through the cell membrane. One way to understand the nature of estrogen hormone is that you can take just a jar of cooking oil and water and shake it up, and it just immediately separates into two phases of water and oil. And that's because the water molecules are very hydrophilic and the oil molecules are very hydrophobic. If you take some salt, just table salt, sodium chloride, it dissolves really well in water, and you add that to that jar of water and oil, and you shake it up, then the oil and water separate. And the salt is said to partition. The salt is going to be in one of the two phases, and it's in the water. It's very, hydrophilic, very easily dissolved in water. There is no salt in the oil. If you do the same thing with estrogen, it partitions into both phases. Almost all of it is in the oil phase. It's very hydrophobic, but there's a small percentage that is always bopping into the water phase. So it's an unusual molecule that's not the first one you learn about in intravio, because it has the ability to be in both phases, but just a low percentage in the water phase and a much higher percentage in the oil phase. And so it's able to go from the blood through the membrane into the cytoplasm that's mostly hydrophilic, and it can end up going right into the nucleus. And so it's unusual. It's an unusual molecule, and the other ligands for nuclear receptors are like that too. They're able to get into the cell so the receptor can be in the nucleus. Much more typically, these signaling molecules don't go into the target cell. They bind to a receptor on the surface. And to make a genomic response, we have to somehow communicate the presence of the signal bound to the receptor into cell membrane into the cell, and in the case of genomic response, into the nucleus. And so that's the whole basis for the PGF story. PDGF doesn't go into the skin cells under a scab, it just binds the receptor on the surface. The PDGF receptor gets activated, it causes an exchange in RAS of GDP to GTP, and that GTP bound RAS binds to this protein called RAF, and RAF is a kinase that will phosphorylate targets. And RAF includes a target called RAF, MEC, and so forth down the line, like that, to ELK, to get into the cell. So that's our talk, and most of the story for us is inside the target cell, and how the target cell is receiving that signal, which often is not, often not, the direct signal inside the cell. So here's just a figure from the start of chapter 16, and it just defines a couple terms because we're in the big field of endocrinology when we talk about cell signaling. And endocrinology includes the study of hormones, and there's a synonym for hormones. Hormones are also called endocrine factors, and they're secreted signaling factors that go throughout the whole body. So estrogen is a good example of a hormone. Endocrine factor is a hormone, any hormone, and it goes throughout the body, and it binds to a receptor. Then there are endocrine factors that aren't hormones that bind locally, and so they're not called endocrine factors, they're called paracrine factors. Also in the big field of endocrinology, and they are local signaling factors. What's that, an example of that? PDGF. PDGF is going to stimulate the cells that are under the scab, local. It doesn't spread throughout the body because we don't want the whole skin to grow, just local stimulation of growth, like that. So lots of examples of both of those, whole systemic growth, like estrogen, or local growth, like the paracrine factor PDGF. And the first part of our book, well, we'll get into it. First part of our chapter, and we'll get into it. Yep. All right. So then, let's just show you, this chapter is divided up into some sections. And the very first section talks about steroid hormones and the nuclear receptor superfamily. Yay. So we've been leaning into that first part. And why would it be first? Because those have signaling factors that are ligands that can go right into the cell. So their stories are relatively simple. Then it goes on to all these others that the signaling molecule doesn't go into the target cell. It binds to a protein on the surface, and we have this signaling pathway into the cell. And there are just a few categories, and they fit into categories like this. PDGF receptor fits into we'll write all this stuff down, but it fits into a section of a group of receptors that are called receptor tyrosine kinases. And so we'll learn about that. And in our bodies, in humans, there are 58 of those. And they're often involved in growth stimulation normally and cancer biology also. So that'll be our main thing. We'll just talk a little bit more about the estrogen receptor in this chapter, but mostly we're going to dive into the receptor tyrosine kinases and what their story is and how the PDGF receptor functions. So you see how that section is organized by in the pathway, right? We have a signaling molecule and the cellular response in the target cell. The focus is, let's organize this based on the receptors that are in the cell membrane. And there are some receptors that function by phosphorylating tyrosines. So the receptor tyrosine kinases, that's how they get activated. So we just bundle those all together. And PDGF receptor is one of them. And in our whole course, we'll just talk about 3 examples of that. and we really will only talk about the PDGF receptor much, because we'll learn about that one really well. But the second one of these that we'll talk about a little bit is the famous insulin receptor. So it's just all of these receptors are just massively important in human health and disease, right? Like that. So we'll really dive into that section. Then, let's see, how is this organized? So this is, just a summary from the, first part of our book, and it goes from one page to the other. Then there's another section that we kind of go back to. Our book deals with it first, and they're called G protein coupled receptors, and they kind of are all grouped together because they use a kind of protein receptor that's called a G protein. And that means they have a similar way of bringing that signal into the cell. And in our bodies, there are 2,000 of these. And they just do all kinds of jobs like that. We won't spend very much time talking about them, even though they're just fantastically, that's how we see, is through G protein coupled receptors. That's how we smell, is through a G protein coupled receptor pathway. Everything like the smell of cinnamon, the cinnamon molecule is binding to a receptor in our, called an olfactory receptor in our noses, that is a G protein coupled receptor. And just on and on and on in our normal health, like that. The people say that something like 70% of all medicines function by regulating a G protein coupled receptor pathway, either upstream of it or the receptor or downstream of it, like that. Just really, really significant. So it makes sense that. And then the last, then there's a section that is kind of a grab bag, right? So there's some really important receptors that kind of have their own pathway, their own sort of story. They're not receptor tyrosine kinases. They are not G protein coupled receptors. And we'll learn about 3 or 4 or 5 of those that are just kind of a grab bag, even though they're super important stories, like that. But it's just how they have to organize the chapter. And this is how people all talk about these pathways too, is they talk about the receptor especially. And then the last section is talking a little bit about broadly about what is going on. What are we talking about here, and what's how's it how are these pathways regulated and stuff like that. So again, a little bit about the the arrestin receptor, nuclear receptors, and then on to the PDGF receptor and stuff like that. Okay, great. All right. So here's that PDGF receptor, and this is a figure from this chapter, but we've looked at it before. It's involved in healing. And it's gonna cause a genomic response, and the genomic response is gonna increase the cell cycle. So we're starting with this example partly because we're already leading into the chapter that's after our first exam, cell cycle, and how we regulate the cell cycle. And so here's the PDGF receptor. It's a receptor tyrosine kinase, and see how it's got those phosphorylated marks there. This is the intracellular domain, that's the extracellular domain, that's the membrane spanning region, like that. So let's just look at that receptor and talk about that for a minute. And so we're talking about these receptor tyrosine kinases, of which there are 58 in us. And they all look like this, where they all are in the membrane. They're transmembrane proteins. They all span the membrane, and they're all in the membrane as monomers. So there's going to be this receptor in the monomer, in the thing, and we can say we're talking about the PDGF receptor, but it's true for all of them, PDGF receptor. It's true for all of them. And then this ligand, this signaling molecule arrives, and in our case, it's PDGF. So here's PDGF. It's released from platelets, platelet-derived growth factor. It's released by platelets underneath a scale. And that causes these two monomers to come together and form a dimer. And when they do, that brings their intracellular domains I should have said, this is the cell membrane that we were talking about here. That brings their intracellular domains in close together, and it activates them, and their intracellular domains are enzymes. So there's an enzyme catalytic site there, and they're kinases. They're going to phosphorylate a target, and the target is surprisingly just the receptor. So these receptor tyrosine kinases have a target when they get activated, and the target is themselves. So they are said to autophosphorylate. And that phosphorylated form becomes the binding site of some other protein. So another protein, and it varies, another protein binds. And carries that further into the cell. So that's just a first look, and our book has figures like this showing that. How does this receptor get activated? It binds the extracellular signaling molecule, and that causes the two monomers to be brought together in a dimer that autophosphorylates and becomes a binding site. The phosphorylated form becomes a binding site for the next step. Okay? So we'll just go back to this, and you can see that's what's happening here. The growth factor, PDGF, or insulin, or many other molecules, causes the receptor monomers to come together and activate their kinase domain, intracellular domain, and it just autophosphorylates. And then it's going to bind to another protein. And in the case of the PDGF receptor, you can see it binds to this GH. And this protein is not very important. It's kind of a failed drawing by the artist. It's really just one protein, and that protein includes a binding domain that's going to bind to the phosphorylated form. And it just says here, it's called a SH2, and that stands for a Sark homology domain. And I just bring that up because you'll see that one of those oncogenic retroviral oncogenes from the 1970s was a protein called Sark. And we're not going to talk about Sark. Sark is a protein that can bind directly to this. So we have a next step, and it varies from one to the other, and it's not a big part of our story. We could pick on it, but it just doesn't bring out a lot of cool stuff. Whereas the next thing is that is going to bind to RAS, and RAS is super cool for learning because it's got an Achilles heel. It turns itself off. So when it's activated, it changes GDP for GTP, and it's active as long as it's bound to GTP, and the way it inactivates itself is it hydrolyzes, it breaks down GTP to GDP. And if you have random mutations, they're likely to cause a decrease, a hypomorphic allele, a loss of function. Decreasing that enzyme activity makes the RAS stay active longer, and its job is just to activate the next step in this pathway. And the next step in this pathway is to bind to RAF, and RAF is a kinase that phosphorylates MEK, and MEK is a kinase that when phosphorylated ERK, and when ERK is a kinase that phosphorylates ELK. And so we run into our first weirdness, which is why are there so many RAS, MEK, ERK? Why are there so RAF, MEK, ERK? They all three do the same thing. They're a kinase that phosphorylates a target, and that target phosphorylates another one. And what's happening there is one of the general things that happens inside the cell. And so we want to put a couple of terms down here. The general term we want to put down here is that this is called the general term for this is it's called a signal transduction pathway. And it's the explanation for how we get the signal to be spread into the target cell. Maybe all the way into the nucleus to change transcription. but we got to get it into the cell and have the cell respond. And the signal transductions vary. We have to learn several of them in this chapter. And one of them is this RAF signaling pathway. And it's activated by RAS, and we see this, and then we see ultimately it's going to make a genomic response, it's going to activate ELK and activate gene expression, including genes like MYC sometimes, like that, okay? So signal transduction pathways. And why are they so weird? You'll see we have to meet some weird ones. This is weird. Why is there so many? They do really two main things, and one of them is they amplify the signal. So this is a perfect example of seeing that signal amplification, because enzymes, when they're activated, they just catalyze the reaction over and over and over. So one activated RAF can activate a whole bunch of MEC. One activated RAS only activates one RAF. There's no amplification there, it's just one-to-one. And we have to have only a few of these molecules get the thing going, make a cellular response to this. So we really need an amplification. And enzymes are a great way to amplify a signal. You know, because one RAF can activate a bunch of MEC, and then one MEC can activate a bunch of ERK, and one ERK can activate a bunch of ELK. So that's a really good example to always remember. There's an amplification story here. And then the second thing that is not so obvious here is called integration. And what that means is that lots of times we have lots of checks and balances on these pathways. So yeah, you're going to amplify a little bit and make a big response, but let's be careful about it. And so lots of times there are crosstalks between pathways where you might need two different molecules to activate a cellular response. And they need to have kind of a combination effect like that. And we'll meet an example of that later in our course. There are lots of examples of that. And so integration means that we take this pathway and also require another pathway. Another pathway might also stimulate it, say yes, or another pathway might block it. And these signal transduction pathways can get complicated because of the integration, like that. Okay? So, the signal transduction pathway. Then, we pick this example because, again, it's cute. It's a genomic response. It's going to cause the ELK to get phosphorylated. ELK's a transcription factor that activates gene expression when it's phosphorylated. There are target genes like that. So we can talk about ELK target genes. There are probably several, I don't know, right? But it means genes that have ELK binding to their cis regulatory region and activating it. And MYC is one of those genes, but there would be others and probably a bunch of others. And so we're used to thinking about that now. The estrogen receptor and estrogen, they have a set of target genes. MyoD, set of target genes, like that, you know? So we could call that, and that would be ELK target genes, but we can also call them PDGF target genes, because that's who's regulating this, right? It's the PDGF story. Goes back to that. And all this stuff is just in between PDGF and the cellular response. You know, the biochemistry in between, okay, like that? All right, fabulous. And so we'll use that example a bunch. And we talked about like how you could see insulin in chicken and humans, like that. Okay, so we're used to that idea, right? Okay, so we go back here. This pathway, there are orthologs of every protein on this pathway in every vertebrate. So, you know, we also can talk about the idea of orthologs, not just in the isolated, you know, insulin type thing, but in the function, right? The function of PDGF is to regulate the PDGF receptor pathway. And so that's another way that, you know, there's just massive evidence for the history of what's happened on our planet with life Because when we talk about an ortholog, it's not just an isolated example. Most of the orthologs interact with other orthologs, right? If a species has a PDGF ortholog, it's going to have a PDGF receptor ortholog, you know, and then if it's got a PDGF receptor, well, how's that working? You know, well, it's going to have that, you know, sarcomology domain protein bind, you know, it's just like that. You know, you've got to have all of those like that. Then another thing about that is that during evolution, we can get that whole pathway. People say we can deploy it. So it could potentially get activated in another setting. And the emphasis here is the PDGF receptor in wound healing. But if you look, that PDGF receptor is expressed in a couple of other places in our bodies as well. So evolution has utilized this pathway in more than just wound healing, like that, yeah. And you know, sometimes it's in early development, and sometimes it's in like the brain or the development of the kidney, like that. So when we're learning about this, you know, we have a focused, you know, introduce this, you know, for now and talk about its role in cancer, but you know, these factors are involved, these steps are involved in lots of different stories in our body, or you know, several different stories in our body, and in every vertebrate, like that, okay? Like that, so just that. Now, you don't have to scramble to write this down, but this is that list of transcription factors that we're talking about and learning about. So just to kind of go through them, you know, MyoD, if you make it, it does its job. Myogena does that too. You make it, they just do their job, like that. Get going, you know, domino gets falling. Then there are ones that are not immediately doing their job. So foxtail, elk, and we'll learn about in this chapter SMADs, like that. And then all of these come out of this chapter. Maybe not heat shock transcription, we've already learned that, but beta-catenin and so forth, and notch, you know, intracellular domain and stuff like that. So through this chapter, we'll include that, and then we're almost done with this. So a big part of our first exam is going to be thinking about transcription regulation and the transcription factors that, you know, regulate gene expression and how those transcription factors are switched on and off, like that. So we're working on a list like that, just to show you that. And again, we'll add to that as we go through this chapter, okay? So here's just an image from the book, right? The book is saying, okay, here's the receptor tyrosine kinase story. It's a you know, big family of genes, our proteins, and our bodies. And, you know, they got that kind of organization to them. And you could draw that on, you know, exam. And, oh, what time does this class end? 10? Huh?
45.
Yeah, phew, man. Yeah, sometimes. All right, so then here it's just zooming. Here's our figure just telling you about RAS. And it's saying like, okay, we're going to exchange the GDP for GTP. Then it's going to be active, go, go, go. And then it's going to get inactivated. And the book describes this and gets a little bit confusing, and I already sort of pre-mentioned that. There's a helper protein that binds to RAS and increases its activity. But it's still RAS that's doing the job. So this is kind of an add-on, not to be confused by. Those cancer patients, they get mutations in RAS. And always remember, it's in the coding region, and it's in the part of the coding region that's going to make the catalytic domain of that RAS enzyme that's going to hydrolyze, going to break GTP down to GDP and turn it off. Always like that. And cancer biology is always like that. When you drill down to the molecular mechanism, it's always just like, that's it. You know, and cancer a lot of time involves multiple different things happening, but part of it is a third of all humans have mutations in RAS. And it's that's the story, is we're affecting that way, and it just causes it to be active too much and saying, yeah, that's why I think we're supposed to be growing, let's grow. And the cell does that. It has a genomic response, and that genomic response leads it into cell cycle, like that, yep, like that. And remember, for our first exam, we're just dealing with 2 of these, and they're here for us now in this chapter. So, you know, we've arrived at a lot of stuff that we've been leading into, and, you know, the MYC oncogene and ELK1, both involved in gene activation and cancer, you know, like that, right? Just an AI overview. So we already know some of this chapter, and we got to rock solid on that, and then build up from there, like that. So we say RAS is a monomeric G protein, and people don't usually say monomeric, they just say RAS. But you'll see that when we talk about the very important G protein coupled receptors, they also activate a G protein, and that story is very different. It's a trimeric G protein. So we're just going to always call this a monomeric G protein just to keep us sorted out. And where does it play a role? In the receptor tyrosine kinases. Does the estrogen receptor ever activate in the RAS? No, right? We've seen enough to know. Nope. It doesn't need anything. It doesn't have any signal transduction pathway. The estrogen goes into the target cell like that, right? So none of this is relevant for the estrogen receptor. And we'll see for the G protein coupled receptors, RAS is not involved in those, right? So we're learning about these receptor tyrosine kinases. It's good learning like that. So let's just look at a couple of highlighted sentences here about that RAS, just to get down to it. And I mentioned before, there are three members of the RAS family, and we just need to know RAS. The reality of it, we'll just know one, but just know that there are three, because they'll refer to all three of them, and they have kind of funny names and stuff like that, right? So, and this is this thing where this projector, and I've forgotten to look at it. The very first day I taught in here this semester, I was like, it's not showing all the colors very well. And you can't really dim this classroom in the front. But I've highlighted on here, yeah. And so I apologize. I went over, I told you I was going to go over, I went over after the first class, and they came over here and looked at it. But I'll just read, I'll try to remember to do this because I'm really focused just on this. So just right here, it says the RTK RAS signaling pathway. So they're just referring to the receptor tyrosine kinase RAS signaling pathway as the most frequently altered oncogenic network in cancer, with almost half of all human cancer tumors displaying mutations in the pathway. So RAS is by far the most common because it's like the Achilles heel of that pathway. It's like, oops, for the reasons we explained, like that. And down here it's naming it's Kirsten RAS and Harvey RAS, and I don't even know the other NRAS, like that. And you'll see that those will come up, yeah, and then it says here, RAS alterations contribute to 20-30% of all human cancers. And then it goes into them. So we don't need to learn the details of the different RASes, but remember how we've already talked about how, well, there's breast cancer, and now do you know that there's ER positive breast cancer versus ER negative breast cancer? So as we go into this part 4, we'll see lots of examples of that, where they'll say, RAS is mutated in 30% of all cancers, but it's not 30% of every kind of cancer, right? I'll just say, do you think that RAS is mutated in human breast cancer very often? No. The breast tissue grows during puberty in response to estrogen, and there could be mutations in any point in life where the estrogen receptor gets activated, estrogen-dependent growth of the breast tissue, right? And that doesn't involve RAS at all, So I'll just speculate that, even though a third of all human cancers are in RAS, probably none of them are breast cancer. They're different kinds of cancer. And so this just drills into this a little bit. They're the three different RASes, and they're prominent mutations in the coding region, in the active site, in those three, but they tend to be different kinds of cancer. And it goes back to what's normally controlling the growth of those cells. And that's what gets weird and abnormal in that cancer, right? So it just says, although KRAS, HRAS, and NRAS share functional similarities, they definitely do. They're all doing that same thing. KRAS, gain of function mutations, tend to occur on the 12th codon, so a specific amino acid, while those in HRAS and NRAS occur on the 61st codon. So some, differences like that. And then they'll say that even the details like that are seen differently in different kinds of cancer. And it's just going back to, well, what's normally working on that? So back up here, it says, KRAS mutations are exceedingly common in pancreatic and adenocarcinomas. So pancreatic cancer, super common, you know? And we are not gonna really, it's not medical school, right, like that. We're not gonna go into it, but just that awareness that, you know, that there's that story expands, right? It's RAS, it's in the coding region, in the active site, but it expands because we have 3 different RASes and they're normally used in different places, just because they are in different parts of our body, right? So that, you know, this KRAS must be used pretty commonly in the pancreas, like that, right? And then it says NRAS mutation more common in melanomas, thyroid cancers, and leukemias, and stuff like that, right? So just a little bit of that, and that's that. And these are just the same thing, and I just highlighted the different parts I would sense I was just talking about. Okay? So that's that. So that's actually just kind of leading into what we're going to talk about, like that. We're doing great. Focusing especially on those receptor tyrosine kinases, and focusing on them like that, you know, like, well, no, what does that mean? And we're going to know how that receptor functions, and then how the steps in the signal transduction pathway function, and learn about each of those to some detail, like that, okay? So now we'll just kind of back up and kind of roll into the chapter again. I think that's good. And so this just shows, for example, at the start of the first section, it just says, like, look at some of these molecules. These are the signaling factors. And signaling factors can be lots of different types of chemistry. Insulin is a little protein. Other signaling factors can be peptides. Others can be steroids, like that, right? And here you see estradiol, and you can see, oh man, it's very hydrophobic, right? That's just our super hydrophobic rings and just a couple hydroxyl groups, like that. You know, like ethanol is got just 2 carbons and a hydroxyl group, and ethanol is already not very soluble in water. And that's got, like, you know, 18 carbons or whatever. So just not soluble in water, like that. And so just showing that, and these are some examples of these ligands for the nuclear receptor superfamily. So just to start, do we need to know these structures? You know, no. But we do want to know that these would have to be getting into the cell. So the structures probably are, the ligands of nuclear receptors are somehow able to cross through the cell membrane. Probably they just have some hydrophobic qualities to them, like that, where they would partition in a jar of water and oil. These guys would all partition partly in water, partly in oil, mostly in oil, like that, yep, okay? And so this is just going into it. You know, it's like, this is easy, look, it's just going into the cell, here's a corticosteroid going in here to bind to the glucocorticoid receptor, nuclear receptor, that's how it gets done, you know, and it's just happy to start that chapter that way, because then it's going to say, guess what, lots of signaling molecules can't get into the cell. So they're going to have a receptor up here on the cell membrane, like that, and then it's going to get complicated. But to start out with, nuclear receptors, yay. And again, we don't have to learn the structure of these or you know, any of the other ligands, because the book has a section about ligands, peptides, proteins, and other kinds of prostaglandins and stuff like that, right? Like that. So then it's talking about nuclear receptors. Yeah, here's some examples of the other signaling molecules like that. And, you know, definitely we're not going to learn the names of like this. And this is not going to be like, you know, draw the structure of acetylcholine. No, you know, so don't think that you need to memorize those structures like that. You need to be aware that there are a variety of different kinds of molecules that can be signaling molecules, and they really boil down to only a few get into the cell, and their receptors are nuclear receptors, and all the rest are on the cell membrane, and that's the rest of the chapter, like that. So really quickly, they were like, well, PDGF doesn't get into the cell, so let's talk about receptor tyrosine kinases. And let's talk a little bit about that, more about that, and let's do this here. Let's see if I have this here. Yeah, so rather than bring it up today, let's just talk a little bit more about this pathway. Let's do this here, where we have a little better zoom in on it. So here's the PDGF receptor pathway, and I'm going to just draw this out, that there is a target gene, and it could be MYC. And so it's got an enhancer, and see how it's showing ALK here, and then it's also showing another protein, like that. So this is an example of an enhancer that binds to 2 proteins, where you need them both to get the job done. It's an example of check and balance, right? Like that, an integration like that of like, well, who activate that serum response element pathway? And it's like somebody else. And so you got to get them both going like that, okay? So we're just going to draw this, and you'll see that that MYC regulatory region sometimes gets activated by ELK, and sometimes other stuff. And so here's just a drawing of MYC, and you'll see we come back here in this chapter repeatedly, because of course MYC has got a promoter, the MYC gene promoter, right? And I don't label these, but you have to label them. And it's got this enhancer that looks like this, right? Where this enhancer is going to bind to phosphorylated ELK. And then it binds to a totally different protein that we'll just add to this story in the last couple things. Another transcription factor called SRF, sorry, SRF. Okay, like that. And you just need them both. And there are going to be other enhancers to bind other transcription factors. There are a variety of different ways that you can activate MYC, like that. And so we're just going to talk a little bit about this SRF. And it goes like this. So a long time ago, scientists learned how to, and this is the first chapter that you'll review for the first exam, is they could take a flask, and this sterile flask in a lab, they can add nutrients, liquid nutrients, and cells, and they can do something called cell culture or tissue culture. They can culture cells in a dish. And I talk about, those USM students that are down there doing an internship at Maine Health Institute for Research. Well, Naomi is one of those students, and do you do cell culture down there? Mm-hmm. A lot? Yeah, because you can grow human cells in a dish, and you don't have to worry about the ethics of studying humans like that. They could be cancer cells, and they could be mouse cancer cells, human cancer cells, any kind of cells, you can grow them in a dish, right? So it turns out that in the nutrients, right, that makes sense, just have some nutrients, and then they're growing in a dish, and there's plenty of oxygen and stuff like that, right? So it's a good, happy thing. And every few days they take them out and they dilute them and, add some fresh nutrients, put them back in another dish, maybe like that, right? So they're growing them. And one of the things that's interesting is that in addition to these nutrients, they add a thing that's like an X factor, like a little extra magic mix. And this surprising magic mix is they'll add, like, in addition to the nutrients, that liquid will be, it's often like 10%, and it's fetal calf serum. Sounds super weird, because it is. And fetal calf serum, so like a baby cow, like an unborn baby cow's serum. And serum is the liquid part of blood. And in a fetus, there's lots of growth. And so that means there are lots of growth factors in the blood. And lots of those can activate a lot of different species of cells. Human cells, mouse cells, whale cells, they're just good. And so they add like that. And if you don't have that, the cells just don't grow. They need that factor. Because in our bodies, in animal bodies, cells don't respond directly to nutrients. They respond to growth factors like this. And so there's a good thing to be aware of, because it's not going to be the nutrients, it's going to be these regulatory factors like that. And that's different than individual eukaryotic cells. Individual eukaryotic cells, those cells respond directly to nutrients. You got enough amino acids, enough sugar and calories and stuff like that, let's grow and divide. So here it's going to be that kind of control. So it's not surprising that in addition to the nutrients, you got to have something else in there to get these cells to grow. And that's this serum. So that's where we're going to get that serum response factor. Because what was shown is you get the cells growing well in a dish, and then you just switch out the liquid to just nutrients with none of this fetal calf serum, and all the cells stop growing. And they stop in a cell cycle at a certain point, and they're waiting for that signal from RAS or RAF, that kind of pathway or another pathway like that. And so they call these serum starved cells. like that, you know? And so then they just take those cells and they just add some serum back and they just all go into the cell cycle. And this is people way back when learning about the cell cycle. And the first thing they start doing is they start copying their DNA. They were right at the start of S phase waiting. And they call that the start site in this cell cycle. Is just at the end of G1 and you're just waiting for the signal and you'll go into the cell cycle into copy your DNA. So then the scientists are like, gosh, this is super valuable because all the cells are doing it and it's a great, you know, controlled environment and we can get them to go, you know, like that, you know? And so we gotta study this. We gotta study out, you know, what goes on when we add this serum. And it must be some kind of signaling molecules that we're adding causing a cellular response. And they're like, I wonder if maybe it's a genomic response or a non-genomic response. So, but let's look at a genomic response and see if we can find new genes being expressed like that, you know? And so they found new genes expressed, and we'll say one gene they found expressed was like MYC. And they're like, oh, I see, you know, the serum is stimulating gene expression that's gonna lead to the cell cycle. So it's, you know, a really good example for us because they're gonna figure this out, right? And so then they're gonna do the methods from the assignment I think is due today, right? We're going to find out what transcription factor binds to the regulatory, cis-regulatory region of MYC. We're going to do a deletion study and just find out where that enhancer is that's serum responsive, right? And they find that, and they find that, and so they find in this drawing, you know, this enhancer that finds ELK and serum response factor. They just called this enhancer a serum response element. You know, good name, right? And they're like, God dang it, we got to find the transcription factor that binds to this. So they just went through those efforts that are in the steps in that assignment about learning how to study transcription factors. So they did a footprinting study like that, DNA footprinting study. Oh, there's a transcription factor bound there, yep, you know. And then they did a gel mobility shift down, say, yeah, we can find there's a, you know, protein that binds that. They did affinity purification of that, you know, and started analyzing it, and they found this protein. They said, let's call this serum response factor. And they're like, yeah, that's a good name for students to learn, right? And so SRF is great, and it's a great example of how we get some integration there. It's not just ELK coming from another pathway that we're not going to learn about is this serum response factor. They got to team up to get the job done here on that MYC, cis-regulatory region. Nice story. Because it combines, these, signaling factors with the receptor pathways with genomic responses, which include the cell cycle in this example, which is our next chapter after the exam. So that's a good way to end it. Then I'll hand back these, and we'll get out of here, and everything's fabulous.